Thank you. Can you guys hear me okay? So I'm Lisa Kitchell. I'm with the Wisconsin DNR, and today we're going to tag team you. Myself, Jesse, and Todd are going to do a watershed management approach to, I'm just going to say, mussels of the Fox River. You can read the whole title as it is. I didn't find out how do I advance. Sorry, should have gotten this technically.
Now right there. Click, click. One click.
On the right?
Single click on the left.
On the left.
On the arrow. Sorry. Right
Sorry.
there. Click.
Okay.
All right.
So, so one of the key pieces I want to bring out, and I want to thank two things. One, I want to thank Josh for his very dynamic talk on the herptiles of Wisconsin, and but now we're going to go back from creepy crawlies back to mussels one more time. And one of the key things that I want you to take away from this is how imperiled our mussels are. Everett did a great talk on introducing y'all to our freshwater mussels, and one of the key pieces is how imperiled they actually are. 70% of the species in North America of our native mussels are listed as imperiled, threatened, endangered, critically imperiled, or vulnerable. And 28 species are already extinct, 17 of which occur in Illinois. So these guys are, they are very vulnerable, and that's the reason there's so much focus on trying to protect and restore these guys out there. You look at that compared to the other taxa, and you see how few that we have. Okay, so the other piece that I want to do, and Everett did a good job of explaining the life cycle of the mussels, is the critical piece is the fish host. So not all fish will do for all mussels. Some mussels will only use one host species. Some will use a whole group of species. So it's really critical when you think of mussels that you don't just think of mussel taxa, but you also have to think of the diversity of fish that are at a site so that they have an appropriate fish host. It's critical because when they get onto the gills of the fish, when they are released from the females, they're these little snappy glochidia. Those glochidia need to insist on the gills of the fish. It's the actual fish blood where they develop all of their internal organs. They're rudimentary, not free-living glochidia. Those glochidia have to get on the gills of the fish. They have to undergo that metamorphosis before they can become a juvenile mussel and drop off. So those are two really key pieces to ensuring the longevity of our mussel species. So, but today we're going to talk about the mussels of the Fox River watershed. It's a watershed of 2,700 square miles. It goes from Wisconsin down to Illinois as well. You know, you've been listening to the Fox River watershed all this time. Again, as was mentioned previously, historically, it was a free-flowing river with primarily prairies and occasional woodlands. And now it's an agricultural watershed with a lot of urban areas and a lot of dams and reservoirs that have completely altered that free-flowing habitat. And that's important to mussel species. A lot of mussel species really prefer that free-flowing habitat because that's how they evolved. It's what brings them their food, brings them their oxygen, takes away their waste. So damming them up and having those reservoirs alters the habitat so it's not as available to, or is preferred by many of the species. So we have a long history of having studied and looked at mussels. We have recorded records all the way from the 1870s up through the early 1900s. J. Eldridge wrote a book on the mussel fishery of the Fox River, and what he was doing was documenting the harvest of mussel shells in the river. That was a very important piece to document, and is used to this day to show what we had for mussel resources back in those days. Okay, I don't know what I did. Go ahead.
It's not you, it's me.
Oh, okay, got it. Sorry. So the first interest in mussels was, well, Native Americans used them for thousands of years, but the first real craze for them was the pearl craze in the early 1800s. And what happened was in Europe, people were not only using pearls for jewelry, but they wanted to wear them and adorn all their clothing with them. That craze came over from Europe because the price of pearls became so high, and people would go out in the rivers here, In the Fox River here, they would wade out, they would rip open mussels and look for pearls. Now you go through thousands, tens of thousands of mussels before you find a good pearl. There are pearls out there, they're just very rare. But because the price of pearl became so high, you could get $1,000, $20,000, $30,000. And in the 1800s, getting that much for a pearl was just phenomenal. So people went out and they just took over the resource and ripped and killed a whole lot of mussels. And so then the interest of that waned. But in the late 1800s, the pearl, I don't know why I keep,
You're fine.
there we go, sorry.
Okay.
The pearl button era came into being. So the pearl button started in the Mississippi River. But that spread to all the Midwestern towns. So all these small rivers had button factories on them. And again, people would just go out, they would wade into the Fox River with their gunny sacks, they'd fill them up and they'd take them to the button factories. And the thing was, it was good money. You could get $3 a day, which in the late 1800s was a good price to get. And a lot of people made a lot of money. There are people that have been reported as having been able to purchase a farm with the money they made from harvesting the shell for button material. So that lasted, but it didn't last for very long in the Fox River because it was so accessible. People were waiting out there. And again, they were collecting all the mussels. So pretty soon there were no mussels to be found. And within 4 years, they crashed the fishery here in the Fox River Basin. So basically it was kind of written off as being there were no mussels, mussels were no longer available in the river. And that was kind of the idea for the longest time. And then occasionally mussels were coming back, but they really weren't being harvested. So back at about 2001, Bob Shanzo, who worked for the Illinois Natural History Survey, said, let's check out and see what mussels are out there. So he went out, he did a statewide, or I should say a whole basin-wide study of the Fox River Basin and its tributaries. And he looked at what mussels were where, and he documented what was there. And the fact that the mussels had really recovered. Given that amount of time being left alone, they had recovered and done a pretty good job. We had lost a few species, but at least there were mussels there. And okay, sorry. I'll try not to do this anymore. So anyway, 20 years later, then Jesse and I and some of their associates from the Illinois Natural History Survey decided to go out and look at a watershed approach to the Fox River mussels and see what was there, see if we had lost species since Bob's surveys that he published in 2004, and see if we'd lost species or how things were, if they were increasing or declining. And that's what I'm going to tag team off to Jesse to do the next report.
All right, as that is getting set up, I'm just going to go with a little backstory of the project itself. So I joined the DNR in 2016, and one of my first projects was to document the distribution and abundance of our 50 species of freshwater mussels. And as I was doing that, I noticed one of the species, rainbow, at the time it was called Velos iris, now it's Cambarunio iris, that had 2 populations recorded since 2000. In the entire state, as mentioned, 42,000 miles of stream in the state, 84,000 intermittent miles of stream, and 15,000 lakes. And here we have 2 populations for 1 species. So I thought that was significant. We had this significant organism, but a lack of support from management. And unlike Aaron Rodgers, we have a solution to keep rainbow in the state and not export it out to New York. So in addition to that, Todd, one of our project partners and soon to be presenter here, he developed the Southeast Wisconsin Mussel Working Group. And here we talked about mussel surveys that have occurred. We collaborate collaborated on project ideas. And he brought up the study, the Shansel study that Lisa just mentioned. And that's significant because that was the first standardized survey methodology throughout the basin. There were some smaller studies in the 50s that used standardized survey methodology, but it was only limited to 10 sites in Illinois. There were older surveys going back to the early 20th century that Lisa documented, but those were just lists. You couldn't do too much with the data other than document the presence of either the shell or the organism and possibly the abundance. So here's this 2004 study, and we were like, hey, let's go replicate that study and see what's going on with populations. From that, identify significant remaining assemblages and identify potential conservation opportunities. So here we developed, we established a federal grant with partners from Illinois Natural History Survey. And the three main objectives were to assess the current status of mussel populations in the watershed, revisiting 96 of Shansel's sites. Evaluate the genetic diversity of rainbow mussel, the Velosa iris, in the watershed. Identifying locations not only in the Fox, but throughout Wisconsin and Illinois. And then propagating and releasing that mussel back into suitable habitat where it either persists or we deem viable for it to be reintroduced. Okay, so quickly going over methodology and site selection. So as mentioned, we wanted to replicate the Shansel study, visiting 96 sites throughout the basin. We wanted to do time searches because Shansel did 2 person-hour time searches separated by 15-minute dives. So that way you could run analysis on catch per unit effort, richness, evenness, diversity. And where there was Velosa iris, we could go back, do quadrat surveys, document density, habitat correlations. And then we also wanted to do targeted species searches because that Velosa iris doesn't just occur in the Fox Basin historically, but also in the Milwaukee watershed, the Rock River watershed, the Wabash watershed in Illinois, and the Illinois River watershed. So as you can see from these 4 different photos, our sites range from an urbanized study. Here you have the Fox River at Waterford. I'm sure most of you are familiar with that location. You had Honey Creek, which was basically a jungle, so you have to search in between all those snags and down trees, because you think of Everett's earlier presentation, how the mussels rely on the host fish. You want to find that suitable area, not just for the mussel, but the host fish itself. Here's a photo from Honey Creek at Bell School Road, and you can actually see the shells laid out. Shallow, nice gravel sand substrate where it is one of our highest diversity streams. Conversely, you have the Muskego Canal, and I don't know if many people have canoed that or done anything there, but we went to the site. We saw what you're seeing now. We saw broken glass at the entry point, and we decided to cross that one off the list. But we did do another site downstream in that same canal and found 2 species. So this grant, we're through year 2 in this 3-year grant. So far we've visited 70 sites throughout the basin and over 5,300 live individuals, 24 live species, 2 species by dead shell only, the hickory nut and the purple warty-back, which is state endangered, and 8 state listed species, either state listed to Illinois and or Wisconsin. Some significant findings were the presence of that targeted species, the rainbow here on the bottom left, ellipse, state threatened, sliver shell, which is on the right there, elk toe, and spike, which is state endangered in Illinois, but not Wisconsin. Here's our basin-wide distribution of sites. Here's a more detailed map over on the right if you haven't seen it. That was developed by Justin and Tom. And that that shows locations of all the sites, but also species richness. The most common species of the 70 sites was the plain pocketbook. If you remember back to Everett's video, we had that plain pocketbook that was attracting the largemouth bass with that darter. That's arguably the most common species in the watershed. Followed by giant floater, which is a postfish generalist. So it attracts everything from shiner minnows to carp to basses. I think it's known for about 40 different host fish. White gill splitter, which is a tolerant of various pollutants, unlike a lot of other species. Creeper, ellipse. And ellipse is notable since it is state threatened. And then there's some photos. Okay, so as mentioned, one of the main deliverables of this project is to compare trends in populations throughout the system. Referencing a lot of these past studies, as mentioned, some of these studies go back to the 1800s, but you can't directly compare because they're just noting species presence for the most part. So I don't think we have a laser, do we? We do? Nice. Should be. Aha, nice. Okay, so these are basically documenting the presence of species. So don't note the increase in richness overall. Note the Matson study in 1957-58, the Shanzel study, and then our study so far. Shanzel documented 27 species. We're right up there with him. And Matson, because he only visited 10 sites in Illinois, that information is also limited. Okay, so far we have 40 sites that we have directly compared to Shanzel. Some of the other sites of the 70, we didn't have access, they weren't easily accessed, or it just wasn't safe to access. Other sites were just outside that 500-meter buffer that we used to do direct analysis, comparing the two. But note that the average species gain was 1.2 species per site, which is significant because going into this project, there was an assumption that population trends were declining because that Shanzel study, one of his conclusions were mussel trends throughout the Fox River Basin are declining, and it's because of urban sprawl. Now, he didn't detail anything beyond that, but we use those terms to hypothesize that trends were going to continue to decline, and we got to identify ways to mitigate that or reverse that trend. But so far, at those 42 sites, there's an average species change of positive 1.2 species. Overall, 5 sites we are seeing decreases in a few sites. Pretty stable or no change, plus minus 2 species at the majority of sites, 25, and then an increase at 16 sites. And if you just look at the map, there's no obvious pattern. It's not like in Wisconsin, species are increasing or decreasing. And as you go down into the watershed, downstream, there's not really an increase or decrease. You'll see closer to the confluence of the Illinois, there's a lot more. It seems like there's an overall increase in species gain, but there's nothing significant yet. Since Lisi, myself, and Todd are from Wisconsin, we highlight Wisconsin findings. If you were at the prep presentation 2 months ago, Alison Stodola, she presented a little more information from the Illinois side of things. So as for Wisconsin, average catch per unit effort, and that's determined by mussels per hour, range from 0. Now, one of those sites was actually at Waterford on the Fox, 236.5 mussels per hour. So almost as fast as you could bag them, you were finding mussels. Richness per site, average 6.5 species, ranging from 0 to 14. Live species richness, I should say. Total richness when you factor in the evidence of dead shell. So if, say, here's a white heel splitter, if we only documented dead shell, we would use that in the total richness analysis, but not live, obviously. So it goes from 6.5 to 9.7 species, ranging from 2 to 17. So at least every site we visited, there was at least 2 species, at least historically there. Evenness. Now this is actually an undervalued statistic, because this will actually help make up the diversity. So if you only have 1 or 2 species that's dominating the system, i.e. zebra, quagga mussels, the viability not only for that species but for that system is hanging, in the balance. It's right on the fringe. So if you have more species, if you have 4, as we did here, or more, only making up about 57% of the catch, that's a good sign that your system is stable. Usually about that 60 to 70% catch rate. That's made up of 3 or 4 species. That's when it kind of changes over to a little more unstable. So those 4 species included pimpleback, pocketbush, mucket, and white heel splitter. And then just to note some exceptional sites, that included Honey Creek, the Fox River in Burlington, actually, right up the road here, right? Riverside Park, which is right over there. That one. All right, thank you. We found 11 live species there, in the McGuanago River, which will be detailed here shortly. 14 of 24 species were reproductive, meaning that they were less than 5. We saw individuals less than 5 years old. You might ask, how do you determine the age? You can actually count the growth rings externally and get a pretty accurate estimate, especially individuals less than 10 years old. So you can see a growth ring here. So this one's only 1, probably 2 years old. And then it starts to fade out, 1, 2, 3, 4, going on 5. And then this one's probably 30 or 40. It's a little harder to accurately estimate on past about 7 or 8 years old. So 14 of the 24 species are known to be reproductive for sure. Outside of the McGuanago, only 11 of the 24 species we've documented young individuals at. All right, species viability trends. So when we pair young individuals with a proportion of live to dead animals and also live individuals per site, we can do a trend analysis to see if populations are stable, increasing, or decreasing. So we have on the left, these species are significantly increasing or at least stable, and on the right, decreasing. Now, if you have a background in mussel ecology or life histories, we would expect the rarest mussels to be decreasing, right? But here we see paper pond shell and fragile paper shell that are opportunistic, and they're usually host generalists. So that was kind of an interesting find. These numbers were all very low, so the statistics weren't conclusive enough to determine, hey, we got to target these individuals for conservation. But based on the information we have, those are the trends. Decreasing or of concern. Otherwise, increasing, you have a larger list. And obviously all these trends vary by site, watershed, and by species. As mentioned, Honey Creek, that was one of our best sites at Bell School Road, I think it was, where we found 14 live species. But if you could just go upstream to, I think, County N, I think we only found 4 species. So those numbers vary just within site to site. All right. All right, as mentioned, the targeted animal, rainbow mussel, the genus changed within this project period. So much for the thought that Latin names are more concrete than common names. Endangered in Wisconsin and Illinois, limited to 1 tributary of the Fox. And the Shanzel study also notes that the 1 tributary, which is the Muguanago, may be the only reproducing population in the entire upper Mississippi basin. Illinois, so far they've found 1 shell from the North Branch of the Nippersink Creek within the watershed. But outside, there's a subwatershed of the Illinois, and then the Wabash watershed, where we were able to collect genetic material sent to Shed Aquarium. Wisconsin, we found live mussels in the Muguanago, as expected, but Todd also found individuals in the White River, right here, just up the road, and the East Branch of the Milwaukee. Obviously outside the Fox Basin, but it's to note that they are expanding or occur outside. Dead shell found at sites on the Fox River proper upstream of Big Bend. So we actually found dead shell at 5 sites north of Waukesha down to Big Bend. So old relic shells, but they did persist for some time in the Fox. And then we collected genetic material to determine the genetic structure of known populations. And that will help us identify genetic unique units. So when we do propagate animals and consider augmentation or reintroductions, we can identify, hey, don't cross these genetically unique populations. So don't bring them from the Muguanago to the Milwaukee or the Milwaukee to the Muguanago because they're genetically different. But it can help us determine if Honey Creek is a suitable area for reintroductions and the genetics within the Fox Basin are appropriate for reintroduction, that means Honey Creek or some of these other sites might be valued for reintroductions in the future. So going back to those maps I mentioned at the start, this just shows the small locations of historically known populations of rainbow mussel. So as note, Muguanago, the brighter red, the more individuals have occurred. This is the East Branch of the Milwaukee River, which is the only other known population in Wisconsin. Now there's some bright red units up here in the Sheboygan watershed, and those have not been verified. They're just kind of hanging out in our database, and me and Lisa are like, I think those are just awkward ellipse records. So we got to go back there and confirm that, or maybe this is a new population as a whole. We're not sure. There's some unverified records in the Rock, in the Ashippun, the Rubicon, and the Scuppernong. We need to go back and verify. We looked at all 3 of those rivers. We did find dead shell, but no live individuals. And then Cedar Creek, which is the other one in the Milwaukee watershed, where we only found dead shell material. So you see a range restriction here occurring in Wisconsin, but thankfully we found them in the white. Hopefully we find more individuals in other bodies of water here in the near future. But it's a similar story in Illinois. If you look over here, in the Wabash Vermilion Basin, we have a couple populations, and they had 1 live individual That was in 2018 in the sub-basin of the Illinois River, where they went back and found 2 more individuals. So historically, they're a lot more diverse and expanded, but definitely concentrated to a few locations. And possibly the only viable population known is the McGuanago. So to expand on that, I'm going to pass it over to Todd.
And while we make that transition, I would like to thank especially Jesse and Lisi. And when I first moved up here and started thinking about where I was going to work and what I was going to do, Lisi had suggested that I work in the McGuanago and that would sort of expand capabilities for the DNR and their ability to keep an eye on that system. I'm incredibly grateful for that, as well as for all of you who have been friends, collaborators, volunteers, and a couple of my students who decided to take off today from their spring break to come out and join us too. So it looks like we're ready to go.
Yeah, I think the arrows are working.
Excellent.
Maybe.
That's good. I hope the arrows are working. I hope they keep working. That would be exciting.
I know.
So as I mentioned, I've been working in the McGuanago River for almost a decade now. And one of the reasons why we've got so much attention paid to the McGuanago River, as Jesse mentioned, is that it may be the only viable rainbow mussel population in the area. So we're spending time and attention to take care of that population and to make sure that we're sort of aware of what's happening with it. And although the McGuanago River system is much, much larger than the area where we've got the biggest rainbow population, we've really focused on the couple of river kilometers between Phantom Lake Dam, so right here, and the confluence with the Fox River, because that's where we find most of the rainbows in the system. So we've spent lots of time in this little boxed area on Highway 83 up to the dam, because that's a really large group of rainbows lives there. That may be our stronghold for the species in many ways. So we've had lots of help along the way, including from many of our audience members. I briefly want to talk about why I'm not reporting on the mussels upstream of the Phantom Lake Dam. So one of the things that's going on is that we just don't have a lot of rainbow mussels from upstream. We're aware of lots of habitat changes that may be preventing the mussels from maintaining a population there. We also heard about the way that rivers get subdivided when we add dams to them. And that may be contributing to the lack of these mussels upstream. So we looked at the database that Jesse mentioned earlier. We've got 37 records upstream of Phantom Lake Dam, and 3 of those are rainbows. They're all from different locations, so we only have 1 record of rainbows from each of those locations. The most recent of those records comes from 2006, and we've done surveys more recently and have failed to locate rainbows above the dam. So all of those things suggest that this is not where we should be putting most of our effort. I briefly want to remind you that Jesse talked about catch per unit efforts. Surveys, that's one way that we can standardize how we know what we know about mussel populations. So when we go in and survey a river, he alluded to the fact that there are a lot of historical surveys that don't give us as much information as we'd like, partially because those surveys don't tell us how much area was searched. They don't tell us how much effort was put into that search. And so we don't know, was this just an uncomfortable site? Was this a site with a bunch of silt? Was this a site when it was storming. And so we don't know how to compare site to site. At least catch per unit effort surveys, we know that people went in, they spent two human hours there, and this is what they were able to find. So most of the surveys that we're following here are either 2 or 4 hour surveys. We've been doing 4-hour surveys and stepping that up to 8-hour surveys in large systems, but that doesn't apply to the Muguanago. So when I look back at our catch per unit effort surveys in the Muguanago, in 2015, we did a fairly comprehensive survey from the Phantom Lake Dam down to the confluence with the Fox River, which is where most of the rainbows have been found. We did those surveys about every 100 meters. So we were able to squeeze in 36 of those catch per unit effort sites. And I just want to lay this out because I've got 4 graphs like this. This end of the graph is Phantom Lake Dam. This end of the graph is the confluence with the Fox River. And in between are counts of mussels. And these are just laid out in distance from the confluence with the Fox River. So this is 1/10 of a kilometer, 2/10 of a kilometer, and so on, all the way up to the dam. So one of the general trends here, and the red represents all the mussels that we found. So the red is total biomass of mussels, total number of mussels. It also, although I'm not showing the data here, it's also reflected in the species richness of mussels that we found. So by the time you get to the Fox in the Muguanago, we really run out of mussels, individuals, and species both. What you probably can't see real clearly is the little blue line at the bottom here. Those are our rainbow mussels. So let me zoom in a little bit and show you those rainbow mussels. So these are just the rainbow mussels. We've gotten rid of all the others. And this is why we focus on that site near the Phantom Lake Dam. This is where we find most of the rainbow mussels. We get a little bit of a drop, and then we get another patch of rainbows, and then we're out. And we don't have rainbows for that lower stretch, that lower kilometer of the river down to the Fox River. So the other way that we survey, which is much more intensive, is a grid survey. And the way that we do grid surveys is based on standardization that was spearheaded within the DNR. And the idea here is that now it's no longer effort, because effort is pretty flexible. Like, it's sometimes really hard to compare effort to effort. Like, it's a stormy day. I don't want to get struck by lightning, I'm going in. It's a cold day. I'm searching quick and I'm getting out, right? So those kinds of differences can affect your outcomes, can affect your measures. So sometimes we search on the basis of area. And so to do that, this is an example of 2 grid surveys that were set up near one another. Separate those by a minimum of 100 meters. And we do those in groups of 3 large 5-meter by 5-meter grids. And within each of those 5-meter by 5-meter grids, we sample quarter-meter segments, quarter-meter quadrats. So here's how 2 sites might be set up relative to one another. And so we randomly place the grids, and then we move upstream and downstream, randomly place another set of grids, and so on. And within those grids, we select a random, usually 25 quadrats within each of those 5 by 5 meter grids. So 5 meters on the side, and then we set up a grid within there that's based on half meter intervals, and we select a quarter of those to sample. And you can see how this sort of looks when we do the survey out into the stream. Here's the big grid set up and delineated with the rope. And then within there, we have quadrats that guide us to where we want fully sampled. And I like this picture because somebody's actually hand excavated that and it pulled out all the substrate from that quadrat, gone through it to see if there are any mussels in it. And just in contrast, remember those are randomly selected, so we may miss a whole strip. We may have some concentrated in part of that grid over another. So results of that survey are laid out exactly like before, and we get this same pattern. More mussels, more richness, upstream, closer to the dam, and then we run out of mussels, we run out of species richness as we move downstream towards the fox. Same thing for the rainbows. We get more of them close to the dam and fewer of them downstream. We also would like to hold on to a little bit of information about the trend in rainbow mussels in the Maguanago. So to do that, we're trying to compare year-to-year grid surveys. And grid surveys take a lot of time, take a lot of effort, take some training, and take some coordination to make happen. So they don't happen every year. They don't happen as frequently as we'd like. This first one is from 2014, and we have another one downstream, just a little bit at one of the first bends in the river, downstream of the dam, that we can kind of compare and look at. So we have, and it's adjusted. That's okay. So at the upstream end of the park where we surveyed, we found one rainbow mussel. We pulled a total of 225 mussels, and that constitutes less than 1% of our catch. There's just not many rainbow there. However, upstream, closer to the dam, we have far more. So we found 21 mussels in this case. That was 7.2% of our total catch. So I think expressing this as percent of total catch helps, because that kind of standardizes it to how many mussels we were able to recover. So in the 2018 survey, and obviously the same thing happened with our formatting, but we were able at Highway 83 to find 9 mussels, which constituted 7.2% of our catch. But we expended a little bit less effort that year, and we're unable to sample the full 25. When we look at the most recent survey, grid survey from 2021, we did 2 surveys, one in that area near the dam, and then one downstream near the canoe launch. And in this case, near the dam, we recovered, and it looks like my numbers, whoops, have disappeared. So it says 12, but those 12 come from this one. And we recovered 22 from just underneath. So those are actually kind of promising in the sense that those numbers are holding relatively steady for our grid surveys. But at the same time, there's a ton of variation within those grids. There's a lot of variation amongst the quadrats. And so I think that we need to consider how we're doing that sampling, especially if we want population trends. There are a lot of challenges in surveying for especially small and relatively rare or low population density mussels. And one is that regardless of who you are, there's usually a bias towards sampling larger mussels. You can get a sense for how to feel those mussels, how to find those mussels, how to recover them, and small mussels kind of disappear. Turns out it's usually easier for people who've been doing this for a while to find small mussels, although that's not always the case. We know that there are detection probability differences between surveys. One of the big problems with detection differences is that in a silty site, you might actually be more able to find mussels because you can move your hand through that substrate and find them, but at the same time, that substrate might be worse for the mussels, might not support them as well. So you can imagine some scenarios where those detectability issues wind up giving us problems with the data. And one of the ways that we can solve that is to estimate detectability directly. We can do things like run mark and recapture surveys, and then based on how many marked individuals we get, we can figure out how hard it was for us to locate those mussels. So we can separately determine how difficult it was to recover those mussels, and then we can use that to standardize between our surveys. But that takes a tremendous amount of effort, and we'd already like to be getting out there more frequently. So one of the things that we're looking at as we move forward is the strategy for how do we do detection trends? How do we do population trends as we continue to try and monitor this particular high-value population? So one of the biggest issues for this population is that it's limited. It's isolated geographically. The Fox River probably limits where those individuals can move via fishes. There's really not a lot of opportunities. The fish run upstream, they hit the Phantom Lake Dam, they go downstream, and they're changing the kinds of habitats that they use as they move into the Fox River. That limits where the fish move. And the mussels as adults, they move, but they don't move a whole lot, and especially seldom upstream. So there may be low density populations out there that we want to connect with this population, or we may not. That's part of why we're doing the genetic work, to try and determine which of those populations is appropriate for us to connect to this. And so we've got a number of challenges in effective monitoring here. The other part of this that I wanted to talk about is how we understand that mussel-host connection. So you've already heard a little bit about that connection, how mussels have to infect a fish host in order to turn into adults and live on their own. And we know that we can introduce mussels to a fish host. We can get mussels that transform into their adult form on those fishes, and we can put them back. So we can do that work in the lab. We know that mussels have predictable host ranges. So there are certain mussels, like our lanceline mussels, that use visual predators, use things like centrarchids. We know that there are groups like the quadriline mussels that use catfishes. So we know that there are groups of fishes that are generally used by certain mussels, and that there's a match between those. So the specificity, so how many fish species a particular mussel can use varies widely. It varies for big groups, big taxonomic groups. It varies for smaller taxonomic groups all the way down to species and maybe below that. And one of the things that we know is that while we can infest fishes with these mussel larvae, there are a whole lot of other things that might determine whether a particular fish is an effective host for a mussel in the wild. So does that fish react to those visual lures that the mussel creates? Does the fish attack those lures when it sees them? We know that the mussel larvae have to actually close on the fish's tissue in order to be able to successfully infect that fish. So if they don't react to that fish, that fish won't be a good host. They have to be able to get through the fish's immunological defenses, physical defenses. They have to be able to grow while attached to that fish host. And so we've already started work with Genoa National Fish Hatchery to do laboratory infestations. They're currently successfully using basses, and larvae have attached to the basses and produced juveniles. Those are almost ready for release. One of the things that we also want to do as we move forward with work on the rainbows is to more clearly understand that mussel-host relationship and understand all those barriers in between having viable larvae and those larvae being able to infest a fish, transform, and drop off. So what we're doing in the lab looks something like this. We have basically the physiological match between fish and hosts. We know that if we put those larvae on a bass, we can get mussels back off of that we can release in the wild. The problem is, in the wild, those mussels and fish have to occur in the same location in the river. Those mussels and fish those mussels have to be able to elicit a response, a behavioral response from the fish. And that response has to be sufficiently effective that it gets the larvae from mom onto the fish in high enough numbers and in close enough proximity that it can they can close on the fish and then can be encysted. So we talk about that sometimes as an ecologically relevant host. So which of these physiologically capable fish hosts are actually the ones that are ecologically relevant in the wild? So hopefully the video, okay. Looks like that video maybe it's working. So we don't actually need the sound, but this is one of the rainbow mussels, and as predicted, is really slow. But you can see her lure. There's a pigmented eye spot, all these little danglies that look very much like crayfish legs, and in fact, an extension that looks very much like a crayfish tail. So she also exhibits behaviors that actually elicit responses from the fish host because she's imitating behaviors as well as the shape. Let's hope I can get back to my presentation now. Here we go. So what are we doing about that? We're looking at behavior. Some of my students at Carroll helped to develop a system, a low-cost system, where we attach imitation GoPros to flower pots. And we've successfully recorded fish interactions with mussels. And that might be able to help us better determine the ecologically relevant hosts, better help us see that essential step in mussel life cycle, getting from mom to the host fishes. So that should tell us a number of things about the biology and the challenge of maintaining that ecological relationship in the wild. So with that, I'm going to hand it back to Lisa, who can talk about a couple of the conservation steps that are happening already and that I alluded to.
Uh-oh. Meanwhile, we got this going on.
It's full.
You want to say something about this one? All right.
So what you're seeing there is the flapping fish lure of the lamsolines, and go ahead and do it to the part 4.
Oh, okay. You want to stop this?
Anyway, and then what Todd was showing was the crayfish lure. And what's really cool is they actually flap it, so it looks like a tail flipping of a crayfish going under a rock. And again, these are predatory fish hosts, so they're keying in on that kind of luring mechanism to get the fish close enough to the mussel. If they don't have a predatory fish host, like their host is something like a darter, they'll often release their glaucidia in egg masses called conglutinants that look like worms or bugs or things like that, so that they, when the fish eats those, then they are, they get on the host.
I think we're down arrow. Down arrow should work.
Oh, thank you.
Maybe.
Got it.
Yeah,
Yeah. Okay, sorry. Okay, so next steps. It's been a long day, overload of information today. It's been great. And I just want to thank everybody for presenting all the great information about the Fox River watershed. So next steps are in Wisconsin, our augmentation of the rainbow. We'll be looking at ways to propagate that as well as monitor, I did the wrong one. To restore historic populations. So we're looking at augmentation of the existing population within the Muckwonago River watershed, but also looking where it was historically occurred and putting some of those mussels back out. In Illinois, they kind of are at a administrative snafu, I'd say. They have to develop a species assessment before. Oh, the screen just did something weird. That's okay. They need to do a species status assessment and then write a recovery plan before they'll be allowed to propagate the mussel in Wisconsin or in Illinois. Sorry. So, and when they do that, then they will then be able to propagate them at the research facility for the Urban Stream Research Center. Right now, we in Wisconsin are using the Genoa National Fish Hatchery. And so we don't have to cross state lines and don't have that impediment. And now
That's her. Boy.
it's not
What
going.
did you do?
I don't know.
Press down.
Yeah.
There you go.
Okay. So that's what I just mentioned, that they have to do an SSA and a recovery plan before they can start doing that. The other thing is the genetic analysis. It's really important for us to get an understanding of the genetic analysis so that we can ensure we have genetic diversity. Jesse mentioned this. We've been taking swabs from all of the rainbows we've been collecting, sending them off to Inua at Shed Aquarium, and he's been doing genetic analysis both within the basin as well as outside of the basin, comparing the rainbow populations from inside and outside. And that way we want to make sure that when we do these propagations, we're not making a bottleneck, but we're ensuring that we have good genetic diversity to go on. And, okay. So where we are propagating the rainbows in Wisconsin is at the Genoa National Fish Hatchery. It's in Genoa, Wisconsin. It's along the River Road at the Mississippi River. I highly recommend if you're going by there, stop in. They're great. They'll show you around and you can see what mussels and fish that they are culturing there. So what we did was we collected gravid females from the Muckwonago River. We took them over to Genoa. They infected 216 bass with those, and they got over 32,000, transformed juveniles from those bass. You don't always get 100%, obviously, survival of those juveniles. So they did have a fair number. In October, we called and they had over 8,000 that had survived. And just recently we called and they had 3,000, close to 4,000, which is not a bad return. Often there's a very low return on the number of juveniles that are initially produced and that survive. So a lot of people ask, what can you do to help the propagation and conservation protection of this resource? One thing is I'd say check out the cool mussel poster over there and just be aware of all the mussels that are out there. There are materials on the tables. You can pick up the mussel booklet, even though it says it's for the upper Mississippi River. It covers everything in the state of Wisconsin. It doesn't cover everything in the state of Illinois. But one of the other things is we have a training day, a volunteer day in the White River. Todd mentioned the White River mussel surveys that they had conducted. We will be having that June 3rd. I would encourage you, there's a sign-up sheet if you're at all interested. Come on out and join us and chase mussels in the White River. It's a fun thing to do. But also, I would say, if you're interested in mussels, check out the map here too that, that Tom created, because it shows you all of that, all the sites that have been sampled, and you could look and see what's in your own backyard and see if you have some records that you might not know about. So it comes back to, people do still say, well, what else can we do? Mussels will recover. They have recovered from that overharvest in the pearl and pearl button craze. Given time, they will restore. We came a long way when the Clean Water Act was implemented in terms of providing good water quality. We still obviously have water quality issues. You were here, if you were here all day, you heard lots of issues, but you also heard lots of ways in which those water quality people are trying to improve water quality. be it looking at pesticides, be it looking at phosphorus, neonics, all of those kinds of things. So there's lots of things still out there, the agricultural cover crops. There's lots of ways that things can still be improved and that you can get involved. Mussels are our sentinels. You can use their presence and the information about them to better inform decision making. And I think that gets at it. I think you guys, if you're at this summit, you're already well aware of water quality. You're well aware of maybe some of the different things that are being done to improve it. I mean, the whole Fox River Study Group, the whole removal of dams, which then restores habitat for these things, the Chain of Lakes implementation plan, all of these things are done to improve water quality. So you can get involved personally if you'd like to as an individual. You can do it on a local basis, on a regional basis. And if you don't want to get out in the water and get wet, you can do it by writing letters or supporting legislation to protect and conserve these guys for the future. We've come a long way. The mussels will recover if they're allowed to, and if we can keep improving water quality, they will keep improving as well. The biggest thing I'd like to say is, if you're interested in mussels, get out there and look for them. Look what's in the stream in your backyard. Look what's in the lake on which you live or that you have a cabin in. It's easy enough to do so. And if they're alive, they don't run away fast. You can catch them. And if they're the shells, then you can take pictures. You can take pictures of them live or dead. In Wisconsin, we have an iNaturalist site where you can report those pictures. We get hundreds of pictures from canoeists that say, or kayakers that say, what is this? And we identify them and tell them. We have a brochure over there that gives our contact information for both Jesse and I. So I encourage you to go out and look. Just see what you got in your own backyard. The good thing is you can look at the shells, you can keep the shells, and you can slowly over time learn your mussels. And with that, I will take any questions, and we save the questions for the end for Jesse and Todd, if you want to come on up.
Right here is the confluence of Honey Creek.
I can't hear.
Confluence of Honey Creek in the White River, right here. The dam here that causes the confluence to impacts the natural flow.
I'm going to give it to you because I can't hear you.
And it's like removing this dam help the rainbow mussel population come up the two streams.
Yeah. Oh, do you want to answer? Did you hear what it was?
Would it impact rainbow mussel?
Would it improve it? The removal of a dam, would that improve the habitat? Is that what you're asking?
Rainbows rely on basses or rainbow darters of all mussels. So if you include it, so host fish distribution is not a limiting factor because largemouth basses, smallmouth basses, they're pretty abundant throughout. It's more of a water quality aspect. If you did remove the dam, you'd open up a lot more of that lotic river flow. Gravel, sandy substrate, which they prefer instead of that lentic lake silty habitat. So if you remove the dam, that'd be great. But obviously there's a lot of other forces at play there. But absolutely. Did you have something to add to that?
And I think the other thing that I would add is that I think there are risks to dam removal too, and I would love to hear somebody better versed on that. But dams retain a lot of things, including that silt that can disrupt mussel habitats. And that's honestly one of the things I worry about for the Muguanago River, is that dam has had some threats to it in the past. And I wouldn't want to see the sediment wind up on that mussel population.
Go ahead, 2 bunches.
Yeah, go ahead.
Kind of a follow on to that, Todd. Can you talk a little bit? That's such a finite population, right? Small 3-kilometer area. Can you talk about age distribution? Are a lot of older animals? And
so the question is, can I talk a little bit about the age distribution of the Muguanago rainbow population? We have small mussels, which indicates recruitment into that population. We have a couple of surveys where we took length records. We did not take length records on this most recent survey, the one where we recovered 66. And I think one of the challenges with analyzing length distribution or ring age distributions is that you need a lot of individuals to get a really clear picture of your age structure in the population. But we have positive signs that we have recruitment into that population. We find young mussels with some regularity.
Right, so the next question then is like, where are other habitats similar? If that's holding on and it's sort of, maintaining, where else for potential restoration, right?
Yeah, and I think one of the places we can look is we can look upstream of Phantom Lake Dam, because we don't know why we don't have rainbows up there. And there are a couple of possibilities. One, it seems to maintain populations at low densities. So we would like to know if maybe that's something that detection probabilities just happen to be right and we miss them, or is that dam preventing their hosts from moving upstream and keeping those populations going? That second possibility is something we can fix with captive propagation, right? We could actually move those upstream. That's also a case where the dam removal might be good, but again, with the caveats about risk. But that's something that's actionable. And that's a challenge that we can overcome or help the mussels overcome. Yeah, go ahead.
Can you comment on, in the region of the McWanago between the dam and the Fox River, can you comment on any changes in water quality and habitat structure within the river, so that the reason why you're not seeing mussels closer to the mouth?
Yeah, so the question is there anything about habitat that we could talk about between the Phantom Lake Dam and the confluence of the Fox River? And we get much, much smaller particle size sediments as we get closer to the Fox. So upstream, we have a nice mix of cobble and sand. As we go downstream, we get more sand, we get more silt. And those are the habitats where I'm not seeing a lot of rainbows. Where it's predominantly sand, we still see rainbows, but when we get a mix of sand and silt, those are habitats, and this is anecdotal. We have some data we can look back at and say that statistically, but my experience is that when we start to get large proportions of silt, we don't see rainbows in those sites. Yeah, of course.
Collectors from robbing them from you. When I was a kid, I would have I lived on the river and I would have been happy to find some of those.
So question is, how do we keep collectors from taking all the mussels? And I think there are a couple of answers there. There's fabulous signage that I was no part of putting in place, but was grateful to see. So there's some education happening directly through outreach. There's some education that's a little bit more passive in terms of signage. The value, the economic value of mussels that Lisi described has declined over time. So there's not the economic incentive. And honestly, I think there's some value in people holding on to the dead shells. I mean, obviously we don't want the habitat to change from that, but people seeing those shells, getting to know those shells helps us to fill in information as they feed it back through platforms like iNaturalist.
Are there signs that say if somebody leaves their car and goes down there that this is illegal or anything?
Yes.
It's illegal to take
Yeah, but people don't
yeah,
know
it's
that.
illegal to take live mussels in the state of Wisconsin. You can collect the shells and everything but the St. Croix and
I'm
Namakogan,
just
and that's,
asking if there's signs there.
yeah, there are.
Yes.
Yep.
Okay, good.
Well, the signs are for endangered mussels. But it is illegal to take them now. See, there were no rules during the harvest eras of the pearl button and the pearl craze. There are now rules and regulations regarding the harvest of mussels. We've closed the commercial harvest industry, and it's illegal for you as a person to take live mussels. You can collect the shell, except for the St. Croix and Namakogan, and that's because it's under Park Service authority where you can't take anything. So you can take those shells for your own personal enjoyment and education.
Can I add to this?
And I just want to be clear regarding pearls. I don't know, some of you may have seen that trending photo on social media of that person who found a white heel splitter on Lake Michigan with like 26 pearls in it this winter or fall. I don't know if you've seen that, but there's a lot of questions behind that photo. Lake Michigan proper does not have an abundance of freshwater mussels, unionid mussels. You may know that they have zebra mussels, and you're not going to find pearls in there either. But between the three of us, how many pearls have we seen? I've seen one in probably 100,000 mussels I've come across.
Yeah.
So very rare.
Yeah, that wouldn't be true.
We had a question over here.
Your genetic studies that are ongoing seem to be focusing on keeping the various populations of the rainbows that you found separated. But you stated an interest in genetic diversity. Those two things seem to be at cross purposes. How does that?
Do you want to take that or you want me to take it? Okay. So one of the important pieces of that genetic diversity is we don't want to find out that we have a genetically unique population in the Fox River that's very genetically different than, say, the ones in the Watershed River. Or if they genetically are very similar, then we could do some mixing it up. But you don't want to find out you have something really unique and then you mess up those genetics by introducing something that has a different genetic makeup. So that's why you want to look at everything that's there and say, and compare those between basin and within basin studies to see what you might, what the differences or similarities might be.
Even though mixing those two populations would, by definition, give you better genetic diversity.
It would, but if you screw up, I shouldn't say screw up, if you mess with that uniqueness of that population, that could be a problem too. You would dilute that unique population and you would lose that population. Or it maybe they're very similar and it doesn't make a difference. We don't know that yet. Ken's in the process of evaluating all of those genetics. He just hasn't gotten it back to us yet. Any other questions? Go ahead.
I have one question.
He's, I can't hear him.
Work on a number of dam removals. In one instance, I can say explicitly that the dam removal was very loss of population. It's very sediment release. But in another instance, there was a series of dams and dam removals that actually restored the sand to the downstream area. It was just open work boulders. And now there was a mixture of sand and ground boulders, which was theoretically would have to be proved. That's my comment. But my question was something observed over the years. We've seen return of some wildlife that was absent for a while, for example, otters. And in the area that I live, there were a lot of the thin shell mussels. But when the otters moved back, they were mittens 3 feet tall. And those are pretty much absent. Do you account for that kind of predation? That's natural, but it certainly would have influenced the mussel dynamics of the population dynamics. Have you seen anything?
So I'm going to quickly just repeat what you said so everybody can hear it. The first part was that some dam removals are in fact really, really problematic for the downstream populations of mussels. Some, whether through luck or planning, are extremely beneficial by recreating the heterogeneity in the habitat. So that was the comment. The question was, what about predation by mammals? And as some mammals have returned, are they still pulling enough mussels to create middens to damage the population? And I guess the first thing that I would kick it off by saying is that those mammals also have a success rate. So as we have more mussels, they'll probably become more successful at getting those mussels, and they may switch to other foods that are available when we don't have as many mussels present. So there is some hope that natural processes will help that. But I'm gonna hand it off to my colleagues and see if we've got more.
Did you have anything else you want to add? No, I think that's one of the things is muskrats, raccoons, otters, they all eat these guys. And I don't think that they have that big a significant impact on the resource compared to what's out there. So I wouldn't worry about those guys. I've seen huge piles of shells and yet found good mussels in proximity to them. So any other questions, or should we now break? Yeah. Yep.
How many were here last year when Director President of the DNR came in? First thing he says, talk to your legislators. Talk to your legislators. The advocacy work that we do by talking to legislators pays for all this stuff. They can't do it. They need to be active with their legislators. During the year, emails, letters, whatever. Speak up to these groups. Because they can't do it. Right now, there's $19.5 billion in new farm conservation coming. We got 8 years to spend it. You need to work with soil awareness to try to get otherwise, you won't get it. One of the other things I want to remind you all is what sells in Middle America. Someday there's going to be a water shortage. Scott's going to be talking about that. Here in the southeastern, Wisconsin, northeastern Illinois, there's going to be a water shortage. Farm conservation can help prevent some of that, help mitigate some of that. Middle America, when they talk about water quality, they're worried about water, they're drinking water for recreation. That's what sells in Middle America. The discussion here, a little tough. That's the issue that legislators are going to vote on. So I encourage you to take a look at your messaging. Work with the legislators, work with your local county boards. I can tell you, I'm also a director of the McHenry Lake County Soil Water District. We get to interface. Okay, I've talked to the Fox Water agencies that have come back to me. They don't work on it in Wisconsin district. Shame on me. Okay. My advice is speak up.
Why don't you give it to Tom? Do you want to wrap up?
The questions?
I think we're pretty much done.
So.
Yeah. And I encourage you to do the YouTube video. Yeah.
Thank you very much, you guys. And I'd like to thank all the speakers and the sponsors and the team that helped put this all together. So thank you very much. I also have a couple announcements. One, mark your calendars, March 14th, 2024. See you here next year. We'll be sending out reminders for that. We're always looking for topics and interesting stories and things about the watershed. So feel free to contact any of us because we try to develop a program throughout the year. Thirdly, if interested, we do have a bartender and the cash bar is open. So we have a little time left here for socializing. And if you can stay around, we're going to have a raffle for the book and a nice thermos. So thank you for taking the time to stay this long. And if you have to leave, so we're doing it. We're collecting name tags, and Ben is collecting those. So those are what we're using to win the raffle. But if you have to leave, please don't forget to drop the name tags off. There's a drop box on the way out by the door. So thank you very much. And on behalf of the whole committee, thank you for attending and making another great day for the summit. So thank you very much.
Say that private and need to get people organized. Yeah. Recreation. Oh, thanks. Thanks for your learning. Awesome story. Here's the guy.
No, let's watch it actually. I know we've been to Lions, I know it's right where the Lions actually do that.
Yeah.
We're that, that training day we need, it's not really-
Yeah, I thought it.
So actually,
I don't know what the downstream of we're all over.
it sounds like we're all trying to get.
I like to read it to the